Okay, got it. So yes, so as Thomas just said, I'm Judith. I've been running a small business which has been focusing in providing artifact-free, reproducible, and quantitative microscopy to users, and been doing this for a number of years now. CERAVI has also been very active in doing quantitative measurements of microscopy equipment, and that's pretty much now known under the ISO standard 21073. So I'm an active member of Quarep Limi, which wants to implement the ISO standard, and Bioimaging North America. So I'm active with these two organizations. And CERAVI has been hired over the years by open science institutions for various reproducibility project in microscopy. So I have a very strong interest in open, reproducible, and fair science. So as Thomas said, this project is being funded by the Molson and Tannenbaum Family Foundation. They provided funds to the OMERO, which is an open science institution here in Montreal, part of the McGill network, and the project itself is under the umbrella of Canada Bioimaging. Okay, I want to start with a quick land acknowledgement. So I respectfully recognize and acknowledge the relationship that the First Nations, Inuit, and Métis people across Canada have with the land that all Canadians live on and enjoy. And I recognize human rights of people who have suffered from systemic racism and discrimination in Canada. And I seek to provide a welcoming, engaging, and safe community for everyone. So, as Thomas said, I'm going to present an introduction on research data management, some of the terminology that is involved in this topic, and I will provide some information about the OMERO instance that we have been running and working on for the last 2 years. So if you don't know already, we're adhering to the FAIR principle. That is, this is a long-standing international movement, and that many scientific communities around the world are adopting. It stands for findable, so making your data findable by both humans and computer software, making your data accessible, and that's usually through secure authorization and authentication process. Making your data interoperable, that is, it has the ability to integrate with other data sets so that, for example, microscopy data sets and genomic data set can work together for the better good of science. And we also want to make data set reusable. So this implies that data set needs to be well described, richly annotated, so that someone can reuse them, possibly with another hypothesis, another tool, another way of, or another purpose. So this is the general description of FAIR principle. And obviously, to achieve FAIR principle, we need to get good research data management in place. And why is this important? Well, first of all, I'm sure that many of you in the audience has been looking for a data set in their life. You know, when we acquire large data set in microscopy, you're going to have data set all over the place, and then you have your Excel sheet analyzing this data somewhere else that's not necessarily linked. So organizing your data is already like a major goal, that a major feature for research data management. You also want to promote quality and rigor. When your data is well organized, You can implement quality checks and make sure that all the necessary information is connected and linked to your data sets. It will foster reproducibility. Other researcher can repeat your experiments and see the same outcome. And you need to provide sufficient information to repeat your experiment. This is particularly true for the step beyond science, like drug and therapy development to cure diseases. And the other important point for a good research data management plan is to share value. When you deposit your data sets in a repository and you have everything well annotated and maintained and curated, it will have a positive impact on your work. It will have a positive impact on scientific discovery, and it's for the good of science in general. In Canada, and that's true for many other places around the world, there's now a data management and sharing policy that is being implemented by the three funding agencies of Canada, CIHR, NSERC, and SSHRC. And essentially, these three funding agencies are now asking institutions to come up with research data management strategy, and the institution need to inform the funders about these strategies. We need to also have data management plans, and again, share this with the funding agencies to describe how about you're getting organized with your data management. And lastly, grant recipients are required to deposit into a digital repository all research data, metadata, and code that support the conclusions that you have when you submit your paper into journals for publication or preprint. So today, I hope I'll convince you that with the help of OMERO, you can actually become compliant to these data management policies. Okay, so again, to illustrate the importance of organizing data, you want the data to be findable and accessible. And if you're dealing with boxes of papers, there are a lot of examples around the world where people have established how to organize data into boxes. But when we come into digital imaging, we have big challenges ahead of us. We'll have information in lab books, on papers, in the drawers of someone, or in the personal computer of a student or postdoc, someone who eventually might leave the lab. We want to associate our Excel spreadsheets with the raw data sets to make sense of all of this. External hard drive, version controls, USB keys. This can be a real challenge for us doing imaging. As I was saying earlier, quality and rigor. Pharmaceutical companies, they know the impact of low quality data and lack of rigor. Back in 2011, there was a first publication, which is showing up on the lower right corner. And they were showing that only 18% of phase 2 clinical trials were successful. And there were numerous publications after that, and the conclusion was always the same, that the major reason for this lack of reproducibility is our inability to report all the information that is necessary to reproduce and document experiments and science in general. Unfortunately, this leads to billions of dollars of research money being wasted every year, every country, all over the world. So down the road, this has an impact on how efficient we can develop drugs and therapies. So more specifically for reproducibility of imaging experiment, this publication here demonstrated that 185 of 240 articles contained original images, and only 17% of the studies passed the minimum requirement for reporting imaging methods. Imaging methods need to are often described very succinctly. There's not enough details, and redoing the microscopy experiment is challenging. But the image processing and analysis method, that's even worse. Reporting in your publication that you use version X of this software is not enough to redo your image processing and analysis steps. Now, that's the downside, but the good side of this is the following. In this study here, this was an international neuroimaging data sharing initiative. They were able to demonstrate that sharing value has a positive consequence for science and for your work. It increased the scale of scientific studies conducted by data contributor. It recruited scientists from a broader range of disciplines. And it, most importantly, it dispelled the myth that scientific finding using shared data cannot be published in high impact journals. So overall, this study brought about over 900 publications, and together they had over 20,000 citations. So sharing a good research data management plan and sharing your science has a good impact on your science. If we now focus more specifically on microscopy metadata, we have three general categories of information, of metadata, that we need to collect when we want to share all the metadata associated with microscopy experiment. The first one is experimental design and sample preparation. The second category refers to the equipment, how you actually acquire the image under the microscope, generate the images. And the last category is image processing and analysis. Experimental and sample metadata, well, that's how you prepare whatever you're bringing under the microscope. And we need to have enough information on sample to interpret the images and support the final conclusions. Here we're talking about the biological origin, the organisms, the part, the tissue section, the organ that we're using. We need to describe how we're actually making the experiment, the date, the protocol, the conditions, the variables that were used in that experiment. And then we also need to talk more about the sample. Is it a section? Is it fixed, permeabilized? What's the antibody we're using, mounting media? And if we're doing live cell imaging, we need to describe how we maintain our sample living and happy under the microscope. So that's information to be gathered for the sample. At the level of the equipment, there are a lot of information, again, that we need to capture. There's information about the microscope hardware, the light source, the filters, the objectives, and so on. And then we need to capture the acquisition, the configuration of all these motorized turret that lives on your microscope, which one was exactly used. Now, the good news is that we're already capturing a number of information there, but we need a lot more. And there are a number of initiatives working on that. And stemming from the publication of the confocal standard back in 2019, we also are now getting organized to collect quality control measurements. So this is to establish how your particular microscope is behaving, and you need to, when you have this information, you actually make conclusions that match what the microscope can really achieve or not. So this is all work in progress. Okay, and then image data analysis, that's the third category, but we need to gather information that will describe, yes, the software, but the operating system, the number of analysis that was run, the number of image cell experiments. And the fourth point here, the detailed analysis workflow, that's super important. If you say, I corrected the background, there's many ways to correct the background. There's many ways to threshold, there's many methods. So this will have an impact on the final output, the final numbers that you get. So we need to record all of that. And we're actually working into making that happen. There are different tools already that can be used to collect, store, and link metadata when you do microscopy. So many of you are not aware of, but there's already bioformat, which is a number of tools developed to make compatible, and so that each manufacturers can use different software to open, visualize, and extract meaningful information out of your images in terms of the metadata. So most of us are already using bioformat, but we're not aware that this is already implemented. Claire's group, and that's mostly the work of Joël. You have a tool there, Methods J2, sorry, the logo did not come out very nicely, but Methods J2 is a software tool that you can use to write your materials and method when you're doing image analysis with Image J, for example. We have Micrometa app. This is an application developed by our colleague Katrina in Boston. And this one allows to not only collect a lot of hardware and configuration metadata, but it also allows you to actually visualize this information as opposed to going to pages and pages of lines of information. Today and tomorrow, we're focusing on OMERO, and OMERO is very central to gathering this information. And we're going to spend a lot of time, obviously, on this topic. But I also want to mention that as a community around the world, we're trying to come up with ways to automatically import images and metadata into OMERO so that in the end, we annotate our science more efficiently with microscopy. Joël, you can meet with Joël, and he's going to tell you more about Methods J2 for creating your materials and method. Okay, so it's just a duplicate. So OMERO is way more than just a database. And what I'd like to show you now is how we're working on integrating various tools into OMERO so that within OMERO, you can become compliant with the research data management policies that the funding agencies are asking you to follow. So again, I'm going to go very briefly into this because we're going to dive much more with our guests here today. But essentially into OMERO, you're going to put your raw data into the database, and can use OMERO to search this database, obviously, but what we all need in microscopy, and this is peculiar for microscopy, is that visualization is super important. Just looking at a bunch of gray levels without their context is useless. So for microscopy, visualizing multi-dimensional data set is really important. And I mentioned this because this is something that the IT people, this is a selling point for IT people. When you tell them that you need a system to visualize the data set, visualization is something that we take for granted, but the IT people do not always understand that it's an important factor for us. So we can do data management, we can do visualization, and we can do quantitative analysis. And the processed data can also be saved into the database. And now you'll notice the yellow flags, and this can all be achieved if we have good metadata annotations with link to our data set. So more detail on the national OMERO instance that we have here available to us here in Canada. And essentially, there is what we call the Digital Research Alliance of Canada. This is a Canada-wide infrastructure for computing science. It's storage, it's advanced computing clusters, and it's interconnected with high-speed connections. We, Canada bioimaging project was prepared. It was Steve and Claire and Thomas were part of that project. And we were granted space on the Alliance, and Steve Ogg installed an instance of OMERO into the cloud. So this is available for all of us here in Canada. And so we've been working in the last 2 years to work with scientists willing and interested in using the database, help them get their data and metadata uploaded into the database. We understand that this is a lot of work, so we would like to automate this process as much as possible for both data and metadata. And because the database lives in the same space as advanced computing clusters, down the road, we want to encourage people to use these tools and resources that are available in Canada. And this is all to foster, in the end, open, reproducible, and fair science. So quickly, I want to acknowledge our colleagues at University of Massachusetts Medical Schools, this Katerina's group. And we've been working heavily with Katerina and Alex, our developer, who is interacting a lot with Alice as well. Alice is our computing person and system administrator for the database. And essentially, we interview with scientists, figure out what they need, how to organize their data set, and how we can automate the process for them for data and metadata upload. One example of what came out of this is that in order to annotate sample preparation and capture the metadata there, we came up with an Excel spreadsheet to collect this information, which at the time of importing to OMERO is reading that spreadsheet and reading more information about the microscope. And this is one of the steps to collect metadata. So in the previous slide, we're focusing on capturing sample preparation metadata. If we use the MicroMeta app application, we collect information from the hardware, and this creates a file that can be read at the time of import. And in the end, into OMERO, we're going to have our data set with a number of key value pairs that describe the sample preparation. And as an attachment or reading directly to the OMERO instance, we can also access the microscope configuration and settings from the database. And lastly, this is still work in progress, but it's just to illustrate that if you do data analysis, you can do them in OMERO, but if you do it in a software that allows you to save your workflow, you can also attach your workflow, your analysis workflow into the database. So the bottom line is that being central onto OMERO, we can essentially have all the microscopy, the sample preparation, and the image processing workflow attached into OMERO. So there's still a lot of work to do because if we want to automate this, we need to come up and agree on a number of terms so that both humans and computers can understand and all agree upon the definitions. And this will greatly improve the interoperability of things. So it's just an example. We know that a confocal microscope, a laser scanning confocal microscope, or a confocal laser scanning microscope, our human brains understand that this is pretty much the same thing, but a computer or software does not necessarily know. So we need to, that's why these things are all super important. Same goes with the Alexa 488 floor 4. We need to all come up with controlled vocabularies for this to happen. And I'm gonna go over this one really quickly, but I do have a document, and if you want to, we can certainly share that. I forgot to print them, but when we work into OMERO, there are a number of things that we can do that will help automation down the road. So, so even if you're not using OMERO right away, when you save your data, there are these little things will help you down the road if you adapt OMERO. So avoid nested folders, one folder into another folder into another folder. That's gonna be challenging in the end. If you can also, I'd recommend that you use the REMB, the recommended metadata for biological images. They're describing, it's like a good start to get organized into controlled vocabulary so that we all name the same thing simultaneously, the same. So have a look at that. Be careful with points. That's not necessarily a problem with OMERO, but down the road, when you get into script and automated analysis, we need to be careful. So for example, if you want to say that you're using your laser at 1.3 milliwatt, well, let's call it 1300 microwatt, and we're going to avoid using a number into that naming file. Here I'm referring to the naming, it's also a naming convention, I should have said. If you get,
catch the eye that you're using you for the micro.
Yeah.
There are computer, there are many keyboard layouts that I can actually create the new symbol.
Yeah, you can create.
Should we use the symbol or should we stick to
We need to, I think it's safer to avoid symbols because we don't know down the road what you'll be using for your script and coding. So
and sometimes, yeah, depending when you do it, the encoding and might change it.
from one country to another.
Yeah,
so it's safer.
yeah, it tends to be the encoding in the file and finally the text might be double down the line instead of reading them the information.
Yeah, these are just practical tips that we discovered which had an impact when we were trying to automate the process.
I want to make sure for everybody understand.
We've seen in many cases that the French accent or German will work quite a lot of it just becomes a headache from the
Yes.
in general, it's nothing to do with it. It's a general computer headache.
Yeah.
Because they tend to consider English as a language of understanding even in computer.
Yeah, yeah. So this is the same reason we say to avoid symbols as much as possible. We need to be careful with dash. except perhaps when you really are truly naming a range, that will be useful. Space can be problematic, so maybe use an underscore. And also this one here is important for OMERO, because if you're using nested folders, we can extract that information with tags in OMERO, but if you only have a number, it will not work. So if you're using, for example, an LSM 780, you might want to put LSM 780 in your name of the file. But if you just do, so LSM 780 can be extracted as a tag in OMERO, but 780 will not be extracted as a tag. So this is why we're saying, in general, if you need to use numbers, add an alphanumerical connection to it.
Just about the ontology, sorry, going back there, with the naming of the proposals and the non-proposals. Is there a working group in Florida that's dealing with that?
Yeah, well, yes, the answer is yes. It's a huge task, and we want to convince more people to join us, because
Which working group?
this is 7.
Yeah.
It's 7, but 7 is collaborating, for example, with 4 and 5 and 2 as well.
For those who don't speak Quora, what are these groups doing?
So in Quora, the first 6 working group are really focused around people generating and testing data sets. So work group 4 is point spread function for with half max. Group 5. Group 4 is color co-registration. And word group 2 is detectors. Word group 1 is light sources. And 3 is XY stage, I think. Illumination uniformity.
We just tell you on the Quora page.
Yeah.
I was just finding out a question think about you before.
Yes, so it's a major challenge. And yes, there are people trying to untangle all of that.
Maybe add what the acronym means for.
QUAREP means quality assessment and reproducibility for images and instrument of light microscopy. So it's essentially taking the confocal ISO standard and breaking it down into these work groups so that we provide user-friendly ways of being compliant to confocal ISO standard 21073, because it's a big document.
My recent answer to the question is, it seems like the ontology needs to come first, because that's really
This is,
early on.
that's right, the ontology needs to come first, and we're having a hard time convincing people of that. So, yeah. And it's boring and it's complicated and it's a huge task, but yes, if we don't take care of that, we might have to redo everything we're doing in 10 years. So, yeah, the last line, it was just an example that we came up on how to name objectives, because often people put the name of the objective in their file, and this is what we're proposing here for naming objectives. So again, I have this document that I can share, and we'll make it available. And now I really want to spend some time here. This is a lot of motivated and dedicated people here that are all working together to make this happen. So we have Alice here, who is our system administrator and computing person that I refer to because I'm not a programmer or coder. Katerina is based in Boston, and she has already established OMERO interoperability with genomic data set for your information. Obviously, we have Claire and Thomas here in the room. Steve, who started this instance. Alex is part of Katrina's lab and Joel is part of Claire's lab. So I think I'm gonna stop right there and take questions if there are any questions. Thank you very much. Yes.
I have a question, mostly kind of about the way that the Canadian instance will work, but also more generally. The way I understand this is working is mostly for groups of, let's say, research groups uploading their own data. I work in a facility. How can I facilitate that, especially knowing that the Canadian Research Alliance space on that cloud is given to researchers and not to, for example, facilities?
So what we recommend that you do if a scientist wants to use the instance, he has to activate his account with the Alliance. So any scientist can get an account. And once he has well, he does actually, no, he doesn't need to have an account. Okay. For the OMERO instance, it's, you communicate with Alice or myself, and we create a group per lab. So each lab gets a group in the OMERO instance. And in this group, there's going to be the PI, and the people in the PI's lab are going to be users associated with this group. And then each user gets credential on top of the PI.
So there's really not much I can do as the facilitator, because I'm hearing automation, and I'm thinking, well,
Yeah.
great, I can get data directly from my machine into a database, because right now I'm not.
This is it. So once you have established your users and groups of PIs and people in the lab, they get their access to the OMERO instance. Then as a core facility, what we can do with you is work out this OMERO, this importer script, which we install on the computer that contains the data. And we can, so as soon as you get an account, people can manually upload data into the database. That's not an issue. But as a core facility, what you could do to help people adopt using OMERO is get into the importer, which we're trying to make it automate the metadata annotation for sample preparation, for microscope, and eventually image processing. So as a core facility, we can work with you to figure out the best way to get the importer running on wherever the raw data set lives. It would be uploaded.
If that is actually connected to that sector. Yes, thank you.
Yeah,
I'm not trying to go in.
We know of your issues and we're ready to go.
So I'm confused about how, I'm not diminishing the size of this app, by the way, but I'm confused as to how the micro-meta app then interacts with the uploader, because we tried installing the micro-meta app and realized it only controls white field and 99% of our usage is confocal.
Yeah.
So until it works with confocal, we're kind of stuck. So do you know where things stand with that?
Look, it's very, the confocal one is almost, I'd say it's 99% ready.
Okay.
Okay, but there were other projects and there were